spleen, the student should be able to elaborate the blood supply of the spleen. Spleen is a very important lymphoid organ which is performing numerous functions in our body. It is one of the largest lymphoid organ of the human body, and it is located in the upper left quadrant of the abdomen, just behind the stomach. The spleen is the only lymphoid organ which is interposed in the blood circulation. The other lymphoid organs are present in the lymphatic circulation. The spleen is performing different functions in the body, and the first important function performed is the filtration of blood. It is helping in removal of the antigenic material and the cellular debris by its macrophages. The spleen has numerous macrophages which are continuously filtrating our blood, removing the antigenic material and the cellular debris. The second important function is it is doing the destruction of old and worn out erythrocytes. The red blood cells who have spent 120 days, they are destroyed by the macrophages which are present in the spleen. As it is a lymphoid organ, it is also an active site of production of activated lymphocytes. The fourth important function, it is helping in storage of blood. As the blood volume decreases due to hemorrhage or due to any other cause, the spleen contracts and releases the blood from its vessels. Hence, it is performing storage of blood. So, four important functions performed by spleen. The first is filtration of blood. Second is site of destruction of old and worn out erythrocytes. Third, site of production of activated lymphocytes. And fourth, storage of blood. So we are going to discuss the structure of spleen under the microscope. When you take a cut section of the spleen, you found that it is covered by a layer of connective tissue, a layer of dense irregular connective tissue which is called as capsule. This lymphoid organ is covered by a layer of connective tissue which is called as capsule. The capsule sends inside septas or trabeculae which divide the parenchyma into incomplete compartments. I am repeating that from the irregular connective tissue capsule, septas enter inside the parenchyma and divide the parenchyma into incomplete compartments. These septas are also called as trabeculae. They divide the parenchyma into two regions, or you can say the parenchyma can be seen to be formed by two different regions which are called as red pulp and white pulp. When you take a cut section of a fresh spleen, the parenchyma of the spleen has two regions, red pulp and the white pulp. The word pulp is used because the parenchyma of the spleen in a fresh specimen appears soft and spongy. Now, this is an illustration, an image histological image of the spleen, in which you can see that the spleen is covered by a layer of irregular connective tissue capsule. This capsule is sending inside septals, or you can say it is sending inside trabeculi, which are dividing the parenchyma into incomplete compartments. When we take the cut section of spleen, the parenchyma appears to be having two different regions. One region which appears red in color is called as the red pulp. Another region which appears lighter in color is called as white pulp. The red pulp is red because of presence of large number of erythrocytes in the capillaries, in the dilated sinusoidal capillaries. And the white pulp appears white in a fresh stain, in a fresh specimen of the spleen, because it lacks the sinuses and consequently it has fewer erythrocytes. So you can see this figure, the spleen which is covered by a capsule, the trabeculi can be seen to divide the parenchyma into compartments, and the parenchyma, also called as the splenic pulp, has two different regions, a lighter region called as white pulp and a darker region which is called as red pulp. Now, what actually is present in the red pulp of the spleen? When you further magnify the region of the red pulp, you will find that the red pulp is composed of splenic cords and splenic sinusoids. The red pulp is composed of splenic cords and splenic sinuses. Now we will discuss the splenic cords. Splenic cords are also called as Billroth cords. These cords are made up of cells, different types of cells aligned together to form the splenic cords. Macrophages, lymphocytes, plasma cells, erythrocytes, granulocytes, and the dendritic cells form the splenic cords. These cells are supported by reticular connective tissue. Look at this image. In this image, you can see that different types of cells are aligned together to form the splenic cords. These cells are macrophages, lymphocytes, neutrophils, dendritic cells, and the plasma cells. The second component of the red pulp of the spleen is the splenic sinuses. Splenic sinuses are actually wide sinusoidal capillaries that lie between the splenic cords. Between these splenic cords, wide capillaries, dilated capillaries, wide lumen capillaries, which are now called as sinusoidal capillaries, are present. These splenic sinuses are lined by fusiform endothelial cells, which are called as stave cells. Now you can look into the image that large gaps are present between the endothelial cells. Another important feature, that endothelial cells rest on an incomplete basal lamina. This is an electron micrograph of the red pulp of the spleen showing sinusoids, red pulp cords, and a large cells which are called as macrophages. The red pulp is supported by the reticular fibers. Now, when you do an HE staining, when you do the hematoxylin and eosine stain, the red pulp exhibits a mixture of eosinophilia and basophilia. The eosinophilia is because of the erythrocytes present in the sinuses, and the basophilia is because of the lymphocytes which are present in the splenic cords. And hence, under the microscope, it appears lighter than the white pulp. Before we move to the white pulp, we should just revise the structure of spleen. The spleen is the largest lymphoid organ. It is made up of lymphoid tissue. It is covered by an irregular connective tissue capsule which is sending inside septas, trabeculae, dividing the parenchyma into compartments. The parenchyma is also called as the pulp because it is spongy in appearance in a fresh specimen of the spleen. The parenchyma is composed of two regions, a white pulp and a red pulp. The red pulp is composed of sinuses and the splenic cords. Now we will discuss the white pulp of the spleen. The white pulp of the spleen consists of typical lymphoid tissue that surrounds and follows branches of the splenic artery. This typical lymphoid tissue forms a cylindrical periarterial lymphatic sheath around each branch of the splenic artery, which is called as central artery. Now look into the figure and try to understand that white pulp is mainly composed of the typical lymphoid tissue. This lymphoid tissue is forming a cylindrical sheath around the central artery, which is a branch of splenic artery, and this sheath is called as periarterial lymphatic sheath. The typical lymphoid tissue, or you can say the periarterial lymphatic sheath, mainly consists of T lymphocytes. At some places within the sheath, there are lymphoid nodules which are composed of B cells. Now remember the anatomy of lymphoid nodules. The nodules are of two types: primary nodules which are not exposed to the antigens, and secondary nodules which are exposed to the antigens and have a central germinal center. Now in the regions where there are lymphoid nodules, the central arteries occupy eccentric position, which is called as a peripheral position, or the best name is eccentric position. Now we'll revise the white pulp. The white pulp is mainly composed of typical lymphoid tissue. The white pulp, this lymphoid tissue in the white pulp surrounds the central artery and is called as PALS, periarterial lymphatic sheath, which mainly consists of T lymphocytes. At places it has secondary nodules. This is an image of the white pulp of the spleen, in which you can see that a lymphoid nodule of the spleen is seen, which is surrounded by the red pulp, the cords, the splenic cords of the red pulp. A germinative center can be seen, as well as the eccentric central artery is clearly visible. So in this image, you can clearly see the white pulp. This is the central artery. This is the periarterial lymphatic sheath, mainly composed of T lymphocytes, and the white pulp also has a lymphatic nodule, which is a secondary lymphatic nodule, as it has a germinative center. Just outside the lymphoid nodule, there is a zone which is called as marginal zone. Marginal zone, it surrounds the lymphoid nodule, and it consists of sinusoids with many macrophages and few lymphocytes. Actually, it separates the white pulp from the main red pulp. When you stain the splenic tissue in hematoxylin and eosine stain, the white pulp stains deeply basophilic because of densely packed lymphocytes. Now, you don't have to get confused. It is simple that when you take a fresh, unstained sample of the spleen, you will find two regions, a red region and a white region. A red pulp having lots of erythrocytes, and a white pulp which is white because of fewer erythrocytes. But when you do the staining of the specimen under the hematoxylin and eosine stain, the white pulp appears deeply basophilic because of densely packed lymphocytes, while the red pulp appears mixed. It takes a mixture of eosinophilia and basophilia. This is an image which is showing you that as the splenic artery enters, it divides and it forms the central artery. This central artery is covered by periarteriolar lymphoid sheath. Outside the white pulp, there is a marginal zone which is composed of sinusoids and macrophages, and the white pulp can be seen to be surrounded by the red pulp. Very clearly in this image, you can see the periarteriolar lymphoid sheath surrounding the central artery. Now in this figure, we are going to revise the structure of the spleen. As we have said, spleen is the largest lymphoid organ, and it is covered by a layer of connective tissue forming the capsule. The capsule sends inside septas, trabeculi, which divide the parenchyma into incomplete compartments. The parenchyma is called as pulp because it is soft and spongy in a fresh specimen. In a fresh specimen, two regions can be appreciated, a red pulp and a white pulp. The red pulp is composed of splenic sinusoids and splenic cords. Cords are made up of different cells, while the sinusoids are wide, dilated capillaries. The white pulp is composed of typical lymphoid tissue, which is which is forming a cylindrical sheath around the central artery. This is an image of the white pulp in which you can see the central arteriole with periarteriolar lymphatic sheath surrounding the central artery. And this is an image of the red pulp in which you can clearly see the splenic cords made up of different types of cells and the splenic sinuses which are made up of wide fenestrated capillaries. Blood supply of the spleen. The splenic artery enters through the hilum and it branches into trabecular arteries. The trabecular arteries, you have to follow the image. The trabecular arteries repeatedly branch and they form the central arterioles. The central arterioles, their diameter is about 0.2 millimeter. They branch and they enter into the white pulp. In the white pulp, they are surrounded by a layer of lymphoid tissue which is called as periarteriolar lymphoid sheath, mostly composed of T cells. And in some regions, it encounters a lymphoid nodule and it takes an eccentric position. The central artery will then further divide and enter into the red pulp to form the peninsular arteries. It will divide to form peninsular arteries. The peninsular arteries will further divide to form arterial capillaries which form the sinuses. Now, some of these arterial capillaries, as you can look into the image, are surrounded by macrophages, or you can say they are surrounded by antigen presenting cells, and they are called as sheeted capillaries. So, again, we will revise. The splenic arteries enter through the hilum, divide to form trabecular arteries. Trabecular arteries divide to form the central arteries. The central arteries are present in the white pulp and they are covered by a periarteriolar lymphoid sheath. The central arteries leave the white pulp, enter into the red pulp, and further divide to form peninsular arteries. Peninsular arteries redivide to form arterial capillaries, which finally form the sinuses of the red pulp. The splenic sinusoids pour their blood into the pulp veins, which unite to form the trabecular vein, which finally unite to form the splenic vein. Now, before we end, we should remember some points. The points to remember are, from the histological point, you have to remember few points. The spleen is the largest lymphoid organ. It is covered by a capsule, a connective tissue capsule. The capsule is sending inside trabeculi, dividing the parenchyma into compartments. The parenchyma is called as pulp because it is soft and spongy and it is composed of red pulp and white pulp. Red because of large number of erythrocytes and white because of fewer erythrocytes. The red pulp is mainly composed of splenic cords and splenic sinuses. The splenic cords are composed of cells, different types of immune cells, macrophages, lymphocytes, dendritic cells, plasma cells, and antigen presenting cells. The splenic sinuses are wide capillaries which are having blood. The white pulp consists of central artery which is surrounded by periarterial lymphatic sheath and